Hey there, my name is Leah Peterson, and I'm going to go over a study that compares the gut microbiome of dogs to the gut microbiome of humans, as well as how the dog gut microbiome is affected by diet. There are two questions that will be addressed, and they are how do the gut microbiomes in dogs compare to the gut microbiomes in humans, as well as how does a dog's gut microbiome respond to a change in diet? So, what do we already know? Well, the gut microbiome of its host is impacted by diet. You can find many studies on this, but one that was cited in this paper was conducted by Lawrence David and Associates, where they were able to demonstrate how quickly the human gut microbiome can change when the diet is changed. We also know that mice have been traditionally used when researching the human microbiome, but according to Fleur Hugenholtz and Willem de Vos, this may not be the best model. The microbiome of humans and mice do share similar genera of microbes, but the abundance is significantly different. Pigs, on the other hand, are closer in phenotype, diet, and their GI tracts to humans, but they are far more expensive to use in studies. Humans, dogs, pigs, and mice all share a similar genetic distance, with the last common ancestor being 97 million years ago. But dogs were domesticated earlier, and for that reason, they have shared their food with their humans for a whole lot longer than if pigs or mice did this. Because of this food sharing, it is believed that their microbiomes are closer in composition to ours. Like people, companion animals around the world are becoming more and more overweight and obese. Alexander German stated that around the world, 22 to 40% of companion dogs are obese. This experiment allowed the authors to explore the differential effects of diet on the microbiomes of overweight dogs. Now, why do we care about any of this? As humans, we love our pets, and as important as it is to keep ourselves healthy, we also want to keep our pets healthy. And when it comes to microbiome research in humans, I believe it is important that we use the most accurate and closest model to our own microbiome. It seems silly to keep using mice if we have found better options. There are two parts to this study. The first was to create a dog gut microbiome gene catalog and then compare it to already existing gene catalogs of humans, pigs, and mice to see which would come out most similar to humans. And secondly, to see how the dog gut microbiome reacts to diet changes to then see if it can translate over into human studies. The first thing the authors needed to do was to create a gene catalog of the gut microbiome of dogs. To do this, they took 64 dogs, 32 Labrador retrievers, and 32 beagles, and then, based on body fat percentage, divided them into an overweight or obese group and a lean or normal weight group. Next, they conducted an 8-week feeding study. For the first 4 weeks, all dogs were fed a baseline diet. At the end of the 4 weeks, fecal samples were taken from each dog as well as body fat percentages. For the next 4 weeks, the dogs were randomly assigned a diet, either a high protein, low carb diet or a low protein, high carb diet, which this one was more closely related to the baseline diet. At the end of the 4 weeks, another fecal sample and body fat percentage was taken from each dog. I just wanted to note here that in order for the authors to avoid confounding effects of changes in the host phenotype, each dog was fed a diet to maintain the weight they came in at. If their weight fluctuated at all, their food was adjusted by 5% up or down to get them back to their starting weight. Also, to control for potential batch effects, the dogs were split into two random cohorts, and one group started a month after the other group. Once the samples were taken, it was time to start preparing the library and sequencing using Illumina. The Nextera XT protocol from Illumina was used to prepare the library. The workflow of this library preparation is as follows. Number 1, tagmentation. It is the process of tagging and fragmenting the genomic DNA. 2, the samples were barcoded using dual index sequencing. The purpose of using dual indexes is to allow you to increase the number of samples sequenced per run and reduce per sample cost. Number 3, amplification was performed in 12 PCR cycles. Number 4, the resulting library was purified using Ampere beads on a robotic platform. Number 5, the quality and quantity of each library was evaluated using capillary electrophoresis. Number 6, the libraries were pooled to ensure proper clusters were formed. To ensure proper clusters were formed, 50 cycles using the Hamilton robot were ran. And then finally, number 7, sequencing was performed on a HiSec 2500 using V4 chemistry. The pool was loaded onto 3 flow cells with pair ends reads of 125 bases in dual indexing. Now we need to build the gene catalog. Reads were first trimmed and then filtered to remove any reads that matched either the canine or human reference genome. Contigs were then assembled for each sample. Genes were then predicted on the reads, and then the genes were clustered at 95% identity for the final catalog. All 129 samples, 2 samples from each dog, with 1 dog contributing 3 samples, were used in the final catalog. This slide shows figure 1A. I'm showing it here because I feel it's a good visual to see the process and numbers from the beginning samples to the final non-redundant genes. I will circle back to this figure shortly in the results section to focus more on the numbers piece. To determine the classification of genes and the functionality of those genes, taxonomic and functional annotations were used. For taxonomic annotation, the dual BLAST least common ancestor approach was used, but instead of using BLAST, DIAMOND was used. DIAMOND is just another classification system, but more sensitive. For each gene, homologs were searched in the UniProt database. If there was a match, that homolog was used as a second homolog, and if that was matched, that region was used as a homolog neighborhood for the initial query gene. The query gene was then assigned as the least common ancestor of the neighborhood. Functional annotation of genes was performed using MoCat2. This is a toolkit for analyzing metagenomic data sets. The Kyoto Encyclopedia of Genes and Genomes, or KEG, orthologous groups were filtered to only include annotations of prokaryotes. These KO groups are groups of genes that perform the same function. Lastly, for the methods section, the total microbial composition of each sample was determined. To do this, MOTULGs were made using a non-redundant database of single-copy marker genes clustered at gene-specific species-level identity cutoffs. The linkage groups, or LGs, were made by correlating marker gene abundances across all canine samples. The result was 228 MOTULGs that contained between 2 and 10 marker genes, accounting for 98% of the total microbial abundance in a sample. Results. The dog gut microbiome gene catalog was created by sequencing 129 fecal samples through Illumina in pair end mode that resulted in approximately 117 million paired end reads, giving a total of 1.9 terabase pairs from all samples. Contigs were assembled from the reads to produce predicted genes. There were over 21 million predicted genes. These predicted genes were clustered together to form the non-redundant gene catalog. In order to make the catalog the non-redundant coding sequences had to have a 95% nucleotide sequence identity to prove the genes belong to the same species. Overall, there was a total of 1.25 million redundant genes identified and added to the dog gut microbiome catalog. Here is figure 1A again. showing how the catalog was created from the initial 129 samples to the 1.25 million genes. Now that the dog gut microbiome catalog has been created, the authors could now compare it to other previously published gut microbiome catalogs of humans, pigs, and mice. Upon comparison of the four catalogs, Figure 1C shows the phylum level distribution of genes. There are 7 categories that are identified in this figure. There are 5 defined phylum: Firmicutes, Bacteroides, Proteobacteria, Actinobacteria, Fusobacteria, as well as other and unannotated categories. The result to note from this figure is the dog and human sections show the most similarity. The human section does show more Bacteroides genes, and the dog section shows more Fusobacteria genes. There's a small area in the human section showing Fusobacteria that can be seen in the actual article, but not in the slide. But overall, they appear most alike. I also want to point out how firmicutes account for the majority of the mouse section, and neither the mouse nor pig contain any Fusobacteria. Figure 1C doesn't account for the difference of abundance of genes or microbes, so a deeper look into the genus level relative abundance was necessary. In this next figure, 1D, we look at how much overlap in gene similarity there are between each of the 4 species. I'd also like to point out that the x-axis has a 19.5% explained variance, and the y-axis has 10.4% explained variance. I also want to note that there are 2 cohorts of human, the light blue taken from MetaHit data, and the dark blue taken from the Human Microbiome Project data. The dog is represented in purple, the pig is orange, and the mouse is green. You'll see that there is a lot of blue on blue overlap, which would make sense because they're both based on human data. Sprinkled amongst the blue in the middle, you start to see some purple. More off to the right, you get the orange from the pig, and then the mouse is hanging out more toward the bottom. The most overlap between species is the human and dog with 26% overlap. Next would be the pig at 11%, and finally the mouse at 4.9%. This figure once again shows that the microbiome of dogs is more similar to humans on the deeper genus level. Next in our results, we're still looking at the similarities between the microbiomes of our 4 mammal species. Figure 1E looks at the data from MetaHit and Human Microbiome Project data and how the human reads compare to the animal reads. You will see on the x-axis are our 4 species, and on the y-axis is the percentage of animal reads that map to the human reads. To start with the human, as we would hope, there was the highest percentage of reads that map to the data. Up next was the dog with 63% of reads mapped to the human reads, followed by the pig with 32.9% reads, and then the mouse with 19.9% of reads mapped to humans. I would like to note that the paper goes a little deeper into this by saying that when mapping animal reads to human reads, the percentages were higher than if you were to map human reads to animal reads, which leads to the dog being at 28% and the pig at 27.2%. There is no explanation to why this is the case, but I did feel like it was necessary to mention it. The last figure in our comparison of gut microbiomes is figure 1F. I like this figure as it shows the number of genes that are shared between each species. When the total number of overlap genes between the dog and human were added up, the result was over 309,000 shared genes. 309,232 to be exact. When the 309,232 genes are up against the 1.25 million genes from the dog catalog, that ends with a 26% overlap in gene pool. If you add up the gene overlap for the pig, you will find there are over 797,000 genes that overlap, but this is out of a catalog of 7.2 million genes, resulting in 11% overlap. The mouse came in at the lowest with over 122,000 genes overlapping a 2.5 million gene catalog, resulting in an 11. Sorry about that. I had to check my numbers. The mouse came in at the lowest with over 122,000 genes overlapping a 2.5 million gene catalog, resulting in a 4.9% overlap. The final number I'd like to mention is the 7 in the middle, where all 4 species come together. This shows that there are over 7,000, so 7,513 genes that are similar among all 4 species. This suggests that there are indeed similar bacteria at the genus and species level of each animal, but most strains contain host-specific genes. All of the subfigures within Figure 1 showed how similarities between species were mapped on different levels using different methods. All of the figures concluded that the dog gut microbiome was most similar to the human gut microbiome, thus proving that maybe the mouse isn't the best model of human microbiome research. So now we're going to switch gears and look at how the dog gut microbiome is affected by diet. To quickly recap, an 8-week feeding study was performed with 64 dogs, 32 Labrador Retrievers, and 32 Beagles, half of which were labeled as overweight or obese, and the other half being labeled as lean or normal weight. In the first 4 weeks of the study, all of the dogs were fed a baseline diet. At the end of the 4th week, a fecal sample was obtained, and the dog's body fat percentage was taken. Each dog was then randomly assigned either a high-protein, low-carb diet or a low-protein, high-carb diet. After 4 weeks on their assigned diets, fecal samples were obtained and body fat percentages were recorded. I'm skipping Figure 2A as it was just a simple visual of our recap, and I will jump right into Figure 2B. This figure shows the phylum-level microbes that were present after the dogs were fed their intervention diet for 4 weeks. The low-protein, high-carb diet, which was closer to baseline, shows the dog gut microbiome composition to be made up of far more bacteroidetes than any other bacteria. The dogs on the high-protein, low-carb diet show a large shift in the number of firmicutes, almost balancing the number of firmicutes to bacteroidetes. The article states that in human studies, the relationship between firmicutes to bacteroidetes correlates with obesity, but the results were conflicting, with some showing a higher ratio, some showing no difference, and some showing a lower ratio. When it came to the dogs at the end of baseline, there was not a significant difference between firmicutes to bacteroidetes ratio, but overweight dogs did have higher bacteroidetes, but after the high-protein, low-carb intervention, both overweight and lean normal dogs showed higher firmicutes to bacteroidetes ratio. This next figure is an additional file, but it is pretty much a visual of what was stated in the last slide. Figure B shows the relative abundance of firmicutes to bacteroidetes at baseline in blue, low-protein, high-carb in green, and high-protein, low-carb in orange. At baseline and with the low-protein, high-carb diet, the firmicutes were much lower than bacteroidetes. But you can see the balancing out with the high-protein, low-carb diet. Figure C shows the ratios as they compare to the diets where the baseline diet has a lower ratio, the low-protein, high-carb has a little higher but not a significant difference ratio, but the high-protein, low-carb diet has a significantly higher ratio compared to baseline. Now to compare at the genus level. The ratio between Bacteroides and Prevotella has been found to be important in human studies where Prevotella is higher in abundance in high-carb diets and Bacteroides is high in high-protein diets. In the dog study, Prevotella was higher at baseline and with a low protein, high carb diet, and Bacteroides was higher with the high protein, low carb diet. These results showed to be consistent with the human results. The next figure is also an additional one, but similar to the last one, showing a visual of the relative abundance of Bacteroides and Prevotella at baseline, with the low protein, high carb diet and the high protein, low carb diet. Prevotella is higher at baseline, then switching to Bacteroides being higher with the intervention diets. Then again, comparing the ratio to the diets. You have the ratio higher at baseline, a little bit lower with the low protein, high carb diet, and significantly lower with the high protein, low carb diet. This next figure, 2D, shows how the highest shift in composition from baseline occurred in overweight dogs on the high protein, low carb diet. The smallest shifts occurred in all the dogs on the low protein, high carb diet, and a moderate shift in the lean to normal weight dogs on the high protein, low carb diet. What I'd like for you to take away from figure 2 is that dogs who were fed a high protein, low carb diet had a greater shift in the types of microbes found in their microbiome compared to the dogs fed a low protein, high carb diet. But overweight dogs showed the greatest shift in overall community composition when fed the high protein, low carbohydrate diet. So now, why such a shift in the overweight dogs? The authors weren't able to narrow this explanation to a single species, but they were able to zero in on 4 genera: Lactobacillus, Prevotella, Streptococcus, and Turicibacter, that all had a significantly higher abundance in the overweight dogs fed the high protein, low carbohydrate diet. This shows that overweight dogs' microbiome was more sensitive to the diet shift from baseline to the high protein, low carb diet. It is also consistent with the view that the microbiome is less stable in overweight individuals than in normal lean individuals. An interesting result of this study is that some taxa became detectable where some became undetectable. An example provided was with Lactobacillus ruminus. At baseline, Lactobacillus ruminus was detected in 22% of the dogs. In dogs fed the low protein, high carb diet, 59% had Lactobacillus ruminus. But in the dogs fed the high protein, low carb diet, there wasn't any Lactobacillus ruminus detected. This actually makes sense, as Lactobacillus ruminus is an immunomodulatory microbe that utilizes complex carbohydrates as a carbon source. Another example from the study was that Intestinobacter bartlettii and the entire genus of Streptococcus was more likely to be found in the dogs on the high protein, low carb diet compared to the baseline or low protein, high carb diet. Because of this, the authors believe these species may be able to be controlled by using prebiotics or by eating food that selectively suppress certain microbe species. This brings us to figure 3a. This figure looks at the prevalence changes of the 3 previously mentioned microbial species before and after the dogs' diets were changed. The first plot shows how Lactobacillus ruminus was present in 22% of the baseline samples, but disappeared with the high protein, low carb diet, but significantly increased with the low protein, high carb diet. The middle plot shows how Intestinobacter bartlettii was quite prevalent, over 60% in the dogs at baseline, but when fed the low protein, high carb diet, its prevalence decreased to around 25 to 30%. And when fed the high protein, low carb diet, its prevalence went up just a tiny bit. The last plot here shows Streptococcus. In dogs at baseline, Streptococcus was prevalent in about 20% of the dogs, but when fed the high protein, low carb diet, its prevalence shot up to about 90%. But when fed the low protein, high carb diet, its prevalence went up just a little bit. The authors suggest that one possibility for this increased prevalence in Streptococcus is because a higher protein content directly benefits these bacteria as proteolytic fermenters. Next, we'll look at changes in the functional composition of the dog microbiome as it pertains to diet changes. To do this, the authors linked the genes responsible in the catalog to KEGG and KZ enzyme classes to get their functional profiles. The strength of the signal was elevated using the MoCat2 toolkit mentioned back in the methods section, which could then, based on their profile, predict the diet the dog was placed on. There were 5 KZ enzyme classes that changed abundance in response to the diet changes. 4 glycohydrolase classes decreased in abundance in dogs on the high protein, low carb diet. This makes sense because glycohydrolase enzymes are responsible for the metabolism of complex carbohydrates. And when the dogs were on the low carbohydrate diet, the need was no longer there for these enzymes, causing them to decrease in abundance. The 5th class of enzymes that had a change in abundance was glycosyltransferase 6. This enzyme increased in dogs fed the high protein, low carb diet. And though its function in bacteria isn't clear, in general, glycosyltransferase enzymes are responsible for forming several different types of glycoproteins responsible for cell-to-cell communication and recognition. Figure 3C shows the 5 enzyme classes that had an abundance change. The first 4 datasets, GH115, GH47, GH48, and GH64, are the glycohydrolase enzymes, where you can see the decrease in the abundance ratio to baseline with the high protein, low carb diet. And the 5th class, the GT6, on the end shows the increase in the glycosyltransferase enzyme ratio in the dogs fed the high protein, low carb diet. So that does it for the results section of this study. Now let's discuss what all this means. When comparing the gut microbiome gene catalogs of dogs, humans, pigs, and mice, dogs may be a better model for research into the human microbiome. The similarities between the dog microbiome and the human microbiome can further the conversation where, if human studies are predictive of results in dogs, why couldn't dog studies be predictive of results in humans? Similar microbiomes have shared benefits to doing dog studies, not only for the dog, but also for the people. Diet really matters. The results of this study have shown that the composition of the gut microbiome really depends on the type of diet that is being consumed. Diet also really matters in weight management. Having a balanced microbial community can factor into weight loss or gain. So what do I think about all this? First, I think this makes a lot of sense. When I mentioned in the background how the domestication of dogs happened pretty early, it makes perfect sense to at least consider the idea that a dog may have a similar gut microbiome to its people. Dogs share food with their owners, they live in the same environment as their owners, they even share life experiences with their owners. Besides cats, there aren't really any other animals that we can do all of that with. So it's not that far-fetched to think that our gut microbiomes would be similar. I'd consider dogs to be much easier to conduct diet research on than people because it can be very difficult to get accurate recall of diet from people. Dogs, you just give them a set amount of food per day, take samples, and collect data. Obviously, there is more to it than that, but it takes a human recall piece out of it. Finally, I think that even though the results of this study have the potential to be beneficial to people, I feel like the dog catalog is lacking. It'd be really hard to put an English Bulldog in the same category as Golden Retriever and to then circle back to how they can compare to humans. I think looking into different biogeography would be beneficial. People and their dogs live in all kinds of places with completely different surroundings. A desert life is completely different than a life in the tundra. There are different plants, different animals, different everything. Also, not everyone has a similar living situation. So to look even into owners that share custody with their dogs like they do their kids. One household could be 100% vegan, where the other is 100% carnivore. I can't imagine their microbiomes would be the same. Plus, it never hurts to have more data. Conclusion. So what do we know? This study showed us that dogs have a more similar gut microbiome to humans than compared to the other commonly used models of mice and pigs. It also told us that the gut microbiome affects the health of its host when it comes to body weight. And overweight dogs had a more sensitive gut microbiome than lean to normal dogs when it comes to the composition and function. And finally, why again do we care? Well, our health is just as important as our dog's health is to us. So, if there's a way to keep us healthy by using our dogs as a model for diet research, then why not? Dogs would do anything for their owners. I don't think they'd mind sharing their gut microbiomes with us if they knew it could help everyone involved to stay healthy. Now, the 5 questions I would like to ask that can help lead discussion is number 1, what could be other reasons why the dog microbiome is more similar to people than the pig or the mouse? Number 2, even though the dog is a better model, should this type of research be done in dogs? Number 3, how could research into the dog microbiome help with obesity research? Number 4, would the microbiome of cats be similar to humans too? Why or why not? And number 5, would feral dogs have a similar microbiome to humans too? References. Thank you so much for your patience with me. This recording thing was kind of tricky. I wish I could have been on video for some of it at least, but I couldn't figure that out. So thank you so much. I hope you enjoyed and learned some stuff about the dog microbiome.